My name is Joseph Wiemels, and I'm a professor at the Center for Genetic Epidemiology at the University of Southern California. My research focuses on the causes of childhood cancer. We explore several risk factors in this series, and here we focus on early nutrition, specifically on the nutrient folic acid and its natural form, folate. Folate is an essential B vitamin with increased requirements during pregnancy. Folic acid is the synthetic, man-made form of folate, and the natural form of this essential nutrient is found in fresh vegetables. Both of these forms of folate provide the primary shuttle service that our body uses to move around methyl groups or carbon molecules within our cells. These methyl groups are used for synthesis of nucleotides and proteins, which are the building blocks for our bodies. These methyl groups are also a primary marker that our body cells use to annotate or label the human genome in early development. While this process looks and sounds complex, its importance to our physiology leads to some straightforward recommendations: good quality nutrition and the use of vitamin supplements during pregnancy for a start. Because this time of life is so important, we will start this presentation in our mother's womb, where all this action takes place. Over 50 years ago, scientists discovered that health and nutrition during pregnancy is extremely important to lifetime disease risk of the child. Scientists found that birth weight is associated with all kinds of diseases, such as diabetes, heart disease, and breast cancer. What is going on during pregnancy with such profound implications? It is this time that our body's physiology is most plastic, or moldable as plastic is, and small changes at this time can change our long-term health history and risk of future cancer. As we get older, our physiology and well-being is hardened or ossified, as stated on the right-hand side of this figure. We all know it is hard to change as we age, and a healthy trajectory starts early and can have impacts on childhood cancer, so pregnancy is an important time where good nutrition can be preventative for future health problems. So, the methyl group that is shuttled by folate is a carbon with 3 hydrogens attached, here called CH3, and this can be attached and removed from cytosine bases in DNA strands. This attachment and removal of methyl groups occurs with sweeping waves over the entire human genome in germ cell and early embryonic development, so clearly an abundance of methyl groups and the shuttle that carries them around, which is folic acid, is crucial at this stage, which ends around the second week of pregnancy. This is a time when folate nutrition and other environmental factors can have profound effects on future health. If we relate this back to the figure on the last slide, we are really talking about the very beginning of the prenatal period. So, how do scientists know that nutrition is so important to this process? It is difficult to study nutrition in people. Indeed, do you even remember what you ate for breakfast? There are, however, a few so-called natural experiments in history when an entire population was subjected to nutritional stress, allowing scientists to follow the health of the population afterwards. One of these is the Dutch Hunger Winter, in which the Nazi blockade of the Netherlands resulted in severe malnutrition for a period of less than a year in late 1944 through to mid-1945. Scientists have followed the health of children born from pregnancies overlapping the starvation period, and it is only those who experience famine in the first 2 months of pregnancies that exhibited long-lasting effects. Here, early gestation starved individuals had lower DNA methylation at the IGF-2 locus compared to their siblings. This effect was present 60 years after the famine. Those that experienced starvation late in gestation did not display any difference in IGF-2 methylation. That data is not shown here. Now, what does this have to do with childhood cancer? Let's look at another contemporary example where we examine DNA methylation right at birth. Here, we start with an interesting natural experiment in Gambia, a country in Africa where nutrition is tightly linked to the season of the year. During the wet season, fresh vegetables are less available, but in the dry season, these are abundant when it is harvest time. Looking at DNA methylation around the genome, one gene was particularly different for children who experienced early gestation in the rainy, low folate time period. This gene, called VTRNA2-1, had elevated DNA methylation indicated here in green. Now, in California, we do not experience large differences in available nutrients by season, since our food distribution system allows access to fresh fruits and vegetables year-round. However, when we look at this VTRNA 2-1 gene in our own DNA methylation data here in California, we find that the pattern of DNA methylation at birth in our leukemia case children looks just like the DNA methylation found in the rainy or hungry season in Gambia. This suggests that children who get leukemia in California appear less well-nourished early in pregnancy, with their epigenome exhibiting signs of folic acid deprivation. Indeed, this provides evidence for a research question, the fundamental question within this presentation. Can early folate nutrition provide a switch towards better health and prevention of childhood leukemia? Fortunately, this question has long been considered with several research groups worldwide, and there is lots of evidence. Let's take a look. Folic acid, the supplemental form found in multivitamin pills, is considered essential during pregnancy and promoted for all women of childbearing years. This recommendation is promoted because of its essential role in the prevention of congenital abnormalities, such as neural tube defects. Several studies have investigated folic acid and childhood leukemia, examining folate during pregnancy and additionally at the beginning of pregnancy, called the periconceptional period. We note in these summaries of these studies that folic acid shows clear protection from contraction of both ALL and AML, providing strong evidence that early folate nutrition is protective for childhood leukemia. The protective impact of prenatal vitamins, including folate, is also apparent for the second most common childhood cancer, brain tumors. Here, we see the strongest impact from folate intake is before pregnancy, which again implicates the preconceptional period as the most critical time period for this protective effect. Let's take another look at what happens to folate and folic acid in a body cell. Folate can enter a cell by way of its synthetic form here at the top of this figure, or in its natural form within fresh vegetables, shown down here. Once entering the cell, folate is shuttled around through various pathways governed by these enzymes, noted here by the blue bars, and natural and synthetic forms of folate will essentially be treated the same. Some of the methyl groups shuttled by folate will eventually end up on the right side of this figure and be used for DNA methylation, which we now know is related to early folate nutrition. Now, every human has these enzymes that control these folate metabolic cycles, but some versions of these enzymes work more efficiently than others, and these different forms are controlled by genetic variation in the human population. Interestingly, investigators have described polymorphic versions of these enzymes and shown that folate levels in the blood can be predicted with some degree of accuracy by genetic variants only, without knowing what a person eats. We can therefore use genetics to measure folate. But that leads to the question, does genetically predicted folate influence leukemia risk? Yes. Included in this figure are several case control studies of genetically predicted folate levels and their relationship with ALL status. The summary odds ratio in the bottom line shows a significant impact on genetically predicted folate. This summary effect shows that folate protects against childhood ALL risk. Now, if genetically predicted folate can prevent childhood ALL, and folate nutrition itself can protect as well, what can both do together? In this figure, we show odds ratios between cases and controls for each unit decrease in folate genetic risk score across study subjects who eat minimal amounts of folate up to high along the x-axis. Interestingly, folate intake has a profound effect in our Latino study group, in which a lower genetically predicted folate level is associated with increased leukemia risk when there is low dietary folate, at the left here. When we increase total folate intake as we move to the right, the odds ratios become neutral, indicating that dietary folate is compensating for the genetic liability. Interestingly, under very high folate levels, the genetic profile may be out of balance, but in the Latino group only. Non-Latino whites here demonstrated increased risk with a weak folate PRS at every level of dietary folate intake. The optimal folate intake level may therefore vary widely, with both too little or too much being a problem. Certainly, folate is an especially vital nutrient, and in the future, our optimal intake levels may be balanced with our unique metabolic traits that are genetically controlled. Finally, we end with some takeaways. First, DNA methylation profiles at birth point towards folate deficiency playing a key role in early development and may increase leukemia risk. Second, epidemiology studies suggest that folate nutrition is a key protective factor against childhood cancer, and this protection may vary among individuals depending on their genetic makeup. Finally, a healthy diet is critical even before pregnancy starts and has lifelong benefits for children.